time to get started with the 11th session of the IEP Annual Workshop. My name is Arnold Ammann. I'll be the session chair for this Delta salmon session. We have a really diverse series of talks, all related to Chinook salmon. And just to remind you, if you've got questions, you can type them into the Q&A box, or you can raise your hand. And at the end of the talk, we'll call on you and unmute your mic, and you can verbally state your question. All right, so with that, the first speaker in our session is Fred Feyrer from USGS. So go ahead, Fred.
All right, thank you, Arnold. I'm going to go ahead and share my screen. All right, is everybody viewing that okay?
Yep.
Wonderful. All right, well, thanks again, Arnold. It's great to be here. Thank you to everyone for coming back after lunch. And I'm happy to be sharing with you today some work that I and this big list of colleagues are doing to trying to address the role of predation on survival of juvenile Chinook salmon migrating through the Delta. So the motivation of our work is that there's this really wide held hypothesis that predation is a key, if not the primary driver of survival of juvenile Chinook salmon migrating through the Delta. There's a lot of lines of evidence supporting this, this idea. Perhaps one of the strongest is this positive flow survival relationship that we see in the system that's pretty well established. It's been looked at for a number of years, and it's held up at a variety of scales under which people have looked at it. But in order to really generate and to implement some effective management actions that are going to address this issue, we really need to understand what the underlying mechanisms of this process really are. And so here's a conceptual model of how this process might work. So what's key here in this conceptual model is how river flow affects stream velocity and also variation in velocity in terms of tides and river channels that juvenile salmon might be migrating through, and how that influences travel time of fish through these migratory corridors. Presumably, then that travel time also interacts with environmental features such as temperature, turbidity, and other variables as well to control or to drive the exposure of these fish to potential predators, and ultimately whether or not they are able to survive and make it through the system successfully. We actually have some pretty strong evidence in support of this particular process here. And so what I'm showing you here on this slide are some data generated from telemetry studies that shows here on the left panel the role of flow on travel time for juvenile salmon migrating through these particular corridors here. And what you can see is that there's pretty much a universal negative relationship between travel time and flow. In other words, more flow reduces the amount of time that fish are spending in these channels, and they're able to migrate through quicker, presumably enhancing their opportunity to survive and reduce interactions with predators. However, as you can see on the right here, we don't have a similar universal relationship with survival. In fact, there's just a few key channels where we actually see a relationship, a positive relationship between flow and survival probability for outmigrating juvenile salmon. And these particular channels are what we call transitionary channels. In other words, these are places where the flow dynamics transition from a unidirectional kind of traditional stream flow environment to a reciprocating tidally driven bidirectional flow environment. So that's a really important feature to keep in mind when we're talking about exposure to predators. And these are those particular channels from that previous slide with those survival data I just showed you. And what we're talking about here is Steamboat Slough, Sacramento River, and Georgiana Slough in the North Delta. And what you can see is that survival probability in these channels varies quite a bit among them. And so Georgiana Slough is where we have the lowest survival probabilities. The highest probabilities are up in Steamboat Slough and Sacramento River. That adjacent stretch of the Sacramento River is where we have, the lowest or the intermediate levels of survival in this case. And so circling back to our conceptual model, this generates a pretty solid foundation for us to further evaluate the effects of predation in a research framework. And so what you'd expect is that if there really is an association between predation and survival in these channels, based upon these data, what you'd expect to see is that the risk to predation is going to have a similar gradient as the survival probability. So what we'd expect to see is the highest predation risk occurring in Georgiana Slough, intermediate in Sacramento River, and highest, or in this case, highest predation risk in Steamboat Slough, or lowest, excuse me. And so that's essentially what we are looking at in our study that I'm sharing with you here today. So this is a progress report of sorts. We're right in the middle of this work, so I don't have a lot of results to share with you, but essentially in a nutshell, what we're really interested in addressing is how many predators are there in these channels and how many salmon do they eat. Eventually, we want to link those data up to the survival estimates. And also, we also are interested in knowing whether or not there's certain habitat features, in particular, diversion pipes in this case, and whether or not these represent hotspots or just locations that help to facilitate predation events. So addressing these questions requires a lot of field work, requires a lot of observations out in the field. And so we've been collecting a lot of field data to support this work. This is what I'm showing you here is kind of the basic approach we're taking to assessing the role of predation in these particular channels here. So what we're doing is we're going out, we're collecting a lot of information on the distribution and the abundance of these predator fishes in these channels and what they're eating as well. And eventually what we want to do is we want to take those data and then model predator population sizes in these channels, how many salmon they're eating, and the extent to which that relates to survival of these fish as they migrate through the system. Our study design, we've got a stratified random site selection that we're utilizing in terms of collecting our data. We're utilizing boat electrofishing. 300-meter transx is kind of our base unit of measure. And importantly, what we're doing, one of the key things that we're doing that's turning out to really help our abundance estimates is that we're taking a number of our sample sites, approximately 15% of them, and doing multiple pass depletion sampling in addition to our single pass widespread sampling to really get at estimating abundances of these fishes pretty well. And it's working out really well. All right, so here's a quick snapshot of some of the data that we're seeing so far in terms of predator abundances in these particular channels. The first thing I'll point you out to for you to take notice of is that the predator composition in these channels in this area of the Delta is comprised of 3 different species of black bass. So largemouth bass, smallmouth bass, and spotted bass. As an aggregate, generally, largemouth bass is the most abundant fish predator-wise that we're seeing in these channels, followed by smallmouth bass. When and where you see a specific species is going to depend a little bit on the location and that specific habitat conditions. But as an aggregate, that's largemouth bass are the dominant predator, followed by the other 2 species. You'll probably notice over here that we did see higher abundances of these predators in 2021 versus 2020. I think that's really mostly an artifact of, we had to shut down pretty early our sampling in the sampling year in 2020. And so we didn't get a full season of sampling. I think that's what's contributing to these higher abundances in 2021. There is an inkling that predator abundances might be highest in Georgiana Slough, but again, we're really early in this project. We're not even finished collecting our field data yet. So this is definitely a work in progress. One thing I wanted to mention here is that the predator field that we're seeing in the North Delta is very different than the predator field in other parts of the system. And so, as I just showed you, the predator field up in the North Delta is comprised of these 3 black bass species: largemouth bass, spotted bass, and smallmouth. Relatively small sizes, small varieties of these particular species. And that's in contrast to what we see in the interior delta, where the predator is largemouth bass. Smallmouth bass and spotted bass really don't exist down in the delta proper, as people like to think of it. And those largemouth bass get really large down in the delta too. So these predator fields are very different. And so when you're considering predation dynamics in the delta, there's no one size fits all. You have to be really mindful of where you're at and what you're looking at and what your predator fields really are, because it's going to really drive the dynamics there. So what are these fish actually eating? We have a little bit of data so far on this, and so this is just a quick plot to show you that the window of observation that we have to look at the diets of these fishes so far. And as you can see, the fish that we've examined so far for diet analysis generally reflect the relative abundance of these species and the size ranges. So we feel like we have a pretty good representative sample so far of the predators in this part of the system. And here's some diet data for you to consider that we've worked up so far. So these are February through April diets, predominantly from 2021. So just keep in mind that these are really, really heavily influenced by drought conditions that we had experienced last year. And so what really stands out to me here is that these fish are really exhibiting no clear transition to piscivory. When and where they eat fish seems to be pretty opportunistic from both these data and some other data that I'll show you in a minute as well. And so what I'm showing you here, these are just very simple, logistic models fit to the presence absence data of fish. And you can see that there's no, there's no clear transition to piscivory like you'd expect, like we've seen in other parts of the Delta, and what you'd expect to be very common from data from other systems as well. So that's pretty interesting. So what kind of fish are these bass eating? So at least last year, they were eating a whole lot of salmon, despite the fact that there were relatively few salmon in the system because most of the fish were trucked around the Delta. We still saw that the majority of the fish that were consumed by these predators were juvenile salmon. So between the salmon prey and steelhead there, the rainbow trout, those two species comprised 50% of the fish that we saw consumed. And as an aggregate, all those other species there comprise the other 50%. One of the things we're also doing is trying to improve our diet characterizations through some genetic work. And this is the work that's being led by Corey and Mark and Wes and you. And what we're doing here is trying to see how we might be able to improve our assessment of how many salmon are being eaten through the use of these genetic tools. And so what I'm showing you here on the left is a plot. These are two plots that Corey generated for his master's thesis at Humboldt State. And this plot here up on the left shows that visually there's at least material still able to be seen up to 120 hours post-ingestion. However, the issue is that roughly at about 6 hours post-ingestion is roughly about the time frame in which you can actually visibly identify a prey with any kind of reliability. You can see that this fish here is pretty well digested. So you're really limited to just a few hours after something gets consumed before you have to start to transition into other tools such as diagnostic bones or genetics tools. And as you might expect, as long as that material is there, you're able to genetically identify it, which is great. So what this is going to do, it's actually going to open up our window of opportunity and our window of observation from about 6 hours up to over 120 hours in reconstructing when and how many juvenile salmon have actually been consumed. So that's going to be super helpful when we get to the modeling aspect of this project. We're also looking at genetics to help us identify not just the presence, absence, but also how many of these fish were eaten. There's been a few papers published on that already, and we're exploring that, that here. And preliminary data suggests it's going to work out pretty well, so we're excited about that as well. All right, so the last thing I wanted to share with you guys is the concurrent study that we've been doing with Cyril and Reed. They're the ones who are leading this particular aspect of it, trying to understand the role of, in this case, diversion structures and whether or not they are features that facilitate predation attacks and interactions between prey and predators. So you guys are probably generally familiar with a lot of the predation event recorder studies that Cyril and Reed and others have been doing, and they applied it here. And what was really great is we were able to integrate it together with what we were doing with our boat-based electrofishing surveys to come up with some really great complementary data that we'll be able to feed into one another in terms of these two particular projects. In addition to using the PERS recorders, they were also integrating some ARIS sonar work, pinpointed on some of the diversions. And so there's a really good set of data here that we'll be able to look at to really understand this process a little bit better. Quick look at a preliminary summary of these data suggests that there's a lot of variability within and among sites in terms of predation events with these observations that were made with the PERS. This is, just like all the aspects of our project, this is all very preliminary. We haven't really gotten too deeply into the analysis yet, but there's definitely some interesting patterns there that we're hoping to look at a little bit more closely in the future as we dive into the data. What has been pretty interesting so far is that, they have GoPros and some other features on these predation recorders to try to identify which predators are actually responsible for the attacks. And what we've seen in those data so far is that it's also, in this case, the black bass species, all three of them, that are really responsible for the majority of the predation attacks that were observed, at least in 2021. So together combined, the black bass accounted for 80% of the predation events, and striped bass about 7% for the observations that could be, accurately, confidently identified. And interestingly enough, we actually, in our electrofishing surveys, caught one of the predators that had consumed one of their salmon on one of their predation event recorders. And so we structured our sampling such that they were doing their work in the evening, and we would go out and sample essentially the same area the next day. And indeed, there was one day we actually caught one of the predators that had consumed one of their salmon. So it's going to be really interesting once we really get a chance to dive into these data and really try to understand this process of how this all works. So again, this was all a very preliminary kind of progress report to share with you all. So, everything's, very first blush, first look at the data that we've been able to collect. A couple initial, very preliminary findings are that, throughout the Delta as a whole, and even up in the North Delta, the predator field is very dynamic. It's going to vary in space and time and in species composition. And one thing that we're seeing that's becoming pretty clear is that these predation events on juvenile salmon are pretty intermittent. They're not constant. It's not this constant pressure that's just kind of applied continuously. They're going to happen when the fish are migrating through the system and when the predators are there. And if you're not looking at the right place at the wrong time, at the right time, you're going to miss it. So that's another reason why we need to really do a lot of field work to capture enough observational data to get this really pinned down. We don't have a lot of information on striped bass. That's a whole nother story that we can talk about for a long time. And so far, at least as of last year, the most common prey is indeed juvenile Chinook salmon. Moving forward, we're still collecting field data this year. In fact, our field crew is on the field today collecting data while I'm giving this talk. And once we have this year's data, we'll be able to really dive deeply into a lot of the analysis and synthesis that we want to do. A few other real quick applications we can take with this work is we can do a lot of habitat growth and population dynamics work with the predators and the prey up in the North Delta. This would be a data set that isn't going to be really heavily reliant on beach stains on boat ramps, so we can really understand the ecology of these fish really well. If you're interested in selective predation, we can, this is something I've thought about, we can characterize the eaten versus the uneaten salmon. And then for folks who are interested in understanding the effects of the North Delta drought barrier, we've got your baseline. We've got 2.5 years now of really good information on what's living in this area and where and what the predation dynamics are. And these are our friends who fund us and keep the lights on while we're doing the work. So I want to make sure I acknowledge all of them, and I'll be happy to take any questions. Thanks a lot.
Thanks so much for that super interesting talk, Fred. If anybody has any questions in the Q&A, we'll have to get to them maybe at the end or later. We've got to move on to our next speaker. Our next speaker is Jeremy Notch, who comes from UC Santa Cruz. He's a NOAA affiliate. He's been working on spring run Chinook salmon for the last 10 years. And he's going to talk to you about some acoustic telemetry studies looking at survival. So go ahead and take it away, Jeremy.
Thanks, Arnold. Here we go. Can you all see this?
Yes, it looks good.
Great, great. Yeah, thank you. So this talk is going to focus on salmon smolt survival through the Sacramento River during the late spring. So as Arnold mentioned, I've done a lot of work with spring run Chinook salmon, and this is kind of the time of year that those juveniles are out migrating. And it's a time of year that conditions drastically change in the river. So we're going to kind of focus on the April-May time period. So just to give you an overview, we're going to look at spring run Chinook salmon in the Central Valley, the current habitat availability, population trends, and the juvenile outmigration strategies across the year. And then take a look at the Sacramento River hydrology during that outmigration period and how natural versus managed flow regime in the Sacramento looks currently. And then take a look at the overall smolt outmigration survival through the river, looking at hatchery smolt survival from a few groups of fish we've tagged going back to 2019 to 2021, and then also the movement rates of those fish through the river. So just a little background about spring run Chinook. You know, historically, they were the most abundant run of salmon in the Central Valley. Essentially, every major tributary to the Central Valley had spring run. Historically, you know, the Southern Sierra, where we have big high mountains, big snowpack. The San Joaquin River had probably the largest run, but after dam construction and you know, water diversions, we currently only have about 3 self-sustaining populations that are recognized: Mill Creek, Deer Creek, and Butte Creek. Although there are, you know, other tributaries that receive spring run occasionally, but these are the 3 kind of core self-sustaining wild populations. And looking at the population trends over time, going back to about early 1990s, you know, right off the top, you can see that Butte Creek by far is sort of driving the spring run populations especially since the early 2000s when a lot of habitat restoration was done in Butte Creek. In-stream flows were improved, dams were removed, and those fish really just came back strong. And Mill and Deer Creek, on the other hand, are relatively lower in abundances, and we had some good returns in the early 2000s and 2010s, but really the last 5 or 6 years, those fish have been struggling. So, you know, one thing about Butte Creek to note is that those juveniles largely avoid a big portion of the Sacramento River. You know, the Butte Creek historically entered the Sacramento River near Colusa, but is now diverted into the Sutter Bypass. And so those fish currently enter the Sacramento River down at Verona. And in addition to that, you know, habitat, rearing habitat is much more productive in Butte Creek with a lot of wetland habitat, and those fish tend to grow quickly and grow quite large, and I think can leave the system relatively earlier in the spring compared to juveniles out migrating from Mill and Deer Creek, which tend to grow take longer to grow. You know, they're spawned and rear in pretty high elevation habitat. You know, Mill Creek has access up to about 5,000 feet. So the water is cold up there. Fish that spawn at that elevation, you know, those juveniles take a long time to grow, and the smolts that outmigrate typically come into the Sacramento River pretty late in the spring. And so looking at the juvenile outmigration timing and sort of strategies for the spring run, we see that there's really two distinct groups. There's a yearling component, fish that juveniles that decide not to leave in the spring, spend the summer months in the upstream habitat, and grow to be quite large and leave in the fall. So typically during, you know, the first few large storm freshets in the fall, but those yearlings will trickle out through the spring. And then the young a year, which are juveniles that leave the system sometimes early, you know, after emerging from the gravel, you know, December, January, and then we see those fish tend to trickle out through the February, March, April timeframe, and as they get larger and become smolts, they're typically leaving the system in about April, May. So a lot of the data that we've collected using acoustic telemetry is really this size range between, you know, 80 and 100, 120 millimeters. So it's a pretty small proportion of the overall life history that these fish exhibit, but I still think it's an important life history. And, you know, as Flora Cordileone, and others have seen, you know, fish that do outmigrate late in the season can have outsized returns as far as the adult escapement. And so juveniles that are outmigrating, you know, from these tributaries and from the Sacramento River in general, historically would typically experience this sort of flow hydrograph. So this represents the Sacramento River. I guess it's the natural Sacramento River hydrograph calculated at Bend Bridge. So it's the Bend Bridge natural gauge, and this is the median flow between 1995 and 2021. And it really just shows that, you know, naturally you have typically storm events in the winter, early spring, and then you have a sort of a receding hydrograph as snow melts, followed by summertime base flows. And this is really what those salmon have evolved to outmigrate in. And currently, this green line shows flows at Bend Bridge, managed flows. And so what these juveniles in the Sacramento River experience today is really the opposite of what is natural, what would be happening if you know, if dams weren't in the picture. So we see that flows throughout the winter and early spring are pretty low. This is a time of year when managers are increasing storage in Shasta Reservoir, so flows are typically low. And then as spring progresses into summer, flows increase to meet agricultural water deliveries, and then sort of peter out again in the fall. And the red line here shows what flows are currently at Wilkins Slough. So again, this is the daily median flow, 1995 to 2021. And flows in the lower Sacramento River at Wilkins Slough are sort of what you would see naturally, downstream. The river does get a bit of runoff from other tributaries. So we see that, water conditions in the winter and springtime are typically good. But until you get to about early, I'd say, I guess, mid-April is really when conditions down there change drastically. So we see flow levels drop from about beginning, mid-April. And this is really due to agricultural water diversions taking place upstream. So really the difference between the green and the red line is how much water is being removed from the system. So smolts that are transiting the Sacramento River are really experiencing quite a pretty drastic change in conditions. And those flows typically reach their lowest values, late April and then May. And then this figure here is overlaying the rotary screw trap catch data at Red Bluff Diversion Dam. And this is just showing the smolt life stage. There's quite a few fry that outmigrate that aren't pictured here, but beginning about March, we see that these smolts begin to outmigrate, and really the peak smolt outmigration that we see at the Red Bluff Diversion Dam rotary screw traps is about early May. And so this time period really coincides with this sort of flip-flop in flows that we see, with increasing flows upstream at Bend Bridge, but decreasing flows downstream at Wilkins Slough. And so this mid-April to mid-May time period is a really important time period for these smolts out migrating through the system, but it's also a time when conditions are changing quite drastically. And water temperature as well. In the blue, we see water temperatures at Bend Bridge, and red, water temperatures at Wilkins Slough. This is 2007 to 2021. And we see that water temperatures upstream at Bend Bridge are relatively stable throughout the year, but they tend to increase quite quickly during that late April to early May time period. So we see flows dropping and water temperatures increasing during that outmigration period. So to sort of look at the survival of fish during this time period, we've been doing a release at Red Bluff Diversion Dam, where we're calling this the Fall Run May release study. It was initially intended to be a pulse flow study that we never got the water for. So we had the tags, we continued to tag and release fish. So we use Coleman Hatchery Fall Run as spring run surrogates between about 80 and 90 millimeters. And fish were tagged at Coleman Hatchery, transported to Red Bluff Diversion Dam, mainly to increase the sample size in the downstream reaches. So in 2019, we had two groups of fish, 250 fish per week released across 2 weeks. 2020, we had more tags, more fish released, about 720 fish across 2 weeks. And then 2021, about 960 smolts released across 2 weeks. And as you can see, we had quite different water year types. 2019 being very wet, 2020 being a dry year, 2021 being critically dry. So we've got a range of water conditions to look at. And just to kind of give a shout out to all the the people, agencies involved in this effort, we have a pretty extensive receiver array that tracks fish, beginning upstream at Redding and going out to the Golden Gate. So the U.S. Fish and Wildlife Service maintains the array from Redding to Colusa. UC Santa Cruz, we maintain the array from Colusa to Verona. USGS maintains the Delta. And UC Davis has been maintaining San Joaquin River South Delta. And then we also maintain the bay. So it's a pretty extensive array. A lot of effort goes into this. In total, over 150 receivers deployed and 50 real-time receivers that transmit detections to the website. You can see that on the bottom. So for this study, for the 3 years that we tagged fish, I selected about 8 sites to look at. We have a lot more receivers deployed, but these 8 sites were consistent across all years and had pretty good detection efficiency. So the reach of interest goes from Red Bluff Diversion Dam to about Knight's Landing. So it's about 200 or so kilometers, and it really encompasses the Sacramento River and all the different habitat types that we see and the changing water conditions. So to estimate the survival for these release groups, I did a simple survival model, which was just release week times reach. So each reach times each release week. So we had 6 different release weeks, and the same for detection probability. So I didn't use any environmental covariates. This was just looking at survival across those release weeks. And here are the per reach survival estimates for those 3 years, with 2019 in blue, 2020 in green, 2021 in red. And these survival estimates are standardized per 10 kilometers because each reach has a different reach length. So if you kind of really squint at this, you can sort of see a pattern, and this is a pattern that we've seen with other tagging studies that we've done in the Sacramento River, which is typically higher survival in the first upstream reach. Typically there's more flow in that upper section of the Sacramento River. And then we see survival tend to decrease around the GCID Irvine Finch, which is kind of the the Chico section of the river. And this is really where water diversions begin to ramp up. And downstream of Chico, survival tends to more or less stay consistent. And then we see survival typically decrease again in sort of the Butte City, Colusa area, and then increase below that. So in general, higher survival upstream, decreasing in the middle Sacramento River, and then it tends to increase in the lower Sacramento River. And so these smolts transiting the river, they experience obviously the different water conditions as water is removed from the system, but also predation can be more of a problem downstream. We do see striped bass coming upstream to spawn around the same time period. So those smolts are going to run into populations of aggregating populations of striped bass at some point downstream. And so these 2 figures are showing survival through the entire Sacramento River. So not per reach, but from Red Bluff Diversion Dam down to Knight's Landing, and comparing survival to flow at Wilkins Slough, which is the lower end of the Sacramento River, and also water temperature in Wilkins Slough. And you can see pretty clearly that in 2019, we had high flows, water temperatures were relatively cool, and survival was a lot higher. So 40 to 55% of those 2 release groups survived the river that year. 2019, water was lower and survival was lower again. And then 2021, which was a critically dry year, we see that survival was practically zero through the Sacramento River, and flows were very low compared to 2019. But there was, one interesting thing that we did see in 2019 when we had high flows. We also had one release group that experienced relatively high water temperature. And so this is sort of an outlier that we've been looking at, that, you can have high flows, but if water temperatures are relatively warm as well, survival may not be what you think. And this figure plots, Wilkins Slough water temperature against Wilkins Slough flow with survival overlaid. And it's, again, it's sort of a linear figure that you can see, when water temperatures are high, flows are low, you're gonna have low survival. But again, this one release group that had, flows upwards of 12,000, water temperature was about 18 degrees, and survival for that group was about 10% lower than the other release group. And so looking at, water temperature at Wilkins Slough during the spring, this figure shows between 2013 and 2020, the water temperature at Wilkins Slough between about mid-March to mid-June. And it shows that, when flows down there are around 5,000 CFS, about half that time during the springtime, water temperature is exceeding 20 degrees Celsius, which is, not ideal conditions for migrating salmon. But we do see in some instances, even during periods of high flow, water temperatures can increase above 20 degrees Celsius. For instance, we had, I think it was 2021, we had water being released from the top of Shasta Dam to basically off-gas it to preserve cold water for winter run. And so, if water is being released off the top of the reservoir, even during high flows, that, temperatures can also increase as well. And finally, looking at movement speed for these 6 release groups, this is travel time in days through the Sacramento River, so Red Bluff to Knight's Landing. And we see that these smolts typically outmigrate relatively quickly. So within 2 to 3 days on average, those fish are out of the system. They're not really rearing anywhere. They're strictly moving downstream. And we see in 2019, those smolts moved more quickly through the system, probably due to the higher flows. And then 2020, fish moved a little bit more slowly. And in 2021, which was the critically dry year, these fish moved the slowest. So, it took those fish an extra couple of days to get through the system. So this really plays into the the exposure of these fish to these smolts, to predation, to pathogens, other negative environmental impacts that may be present. Basically, if there's more flows, those fish are going to move through the system quicker and have less risk overall.
Okay. Hey, thanks a lot, Jeremy. We'll have to get your conclusions later. In the interest of time, we need to move to the next speaker. And that's going to be Jason T. Magnuson and Daniel Schlenk of UC Riverside. And in the interest of time, we'll just go ahead and jump into their talk. Thank you.
Thank you.
So this talk is going to be focused on some recent studies that we've conducted looking at how to characterize overall neurotoxic as well as neuroendocrine effects of bifenthrin to salmonids and the further influence of different environmental factors associated with climate change, such as temperature. And it's coming from an approach of how we can relate several different omic profiles, such as transcriptomics, lipidomics, and metabolomics, to different apical endpoints. In California, there's more than 190 million pounds of pesticide active ingredients applied annually. And over 1,000 rivers and streams are considered to be impaired, 290 of which are impaired considering pesticide use. Since the early 2000s, there's been a sharp decline in several pelagic fish populations, such as the delta smelt, longfin smelt, striped bass, and threadfin shad. And this is also where we start to see the increased use of several pyrethroid pesticides. And so this is showing increases in pyrethroids detected in predominantly agricultural fields, urban runoff areas. And again, this is starting to increase from the early 2000s, where we start to see some of these pelagic fish decreases. Within the delta, bifenthrin, which is a pyrethroid pesticide, we see it is the most frequently detected pesticide, predominantly in urban runoff areas, and also the most frequent to exceed EC50 and LC50 of values for Hyalella species. In a recent report, it was found that bifenthrin was present in 51% of samples taken, and in predominantly agricultural areas, it was shown that the concentrations of bifenthrin detected were above those that exceeded a chronic toxicity to invertebrate species. And in predominantly urban sites, the concentrations found were above those that induce chronic toxicity to invertebrates, but also some fish species. Bifenthrin is the most frequently detected pyrethroid in river sediments in North America as well as Australia, and is known to bind to voltage-gated sodium channels and dysregulate calcium homeostasis in neurons, and it's highly hydrophobic. Additionally, though, we have the threat of climate change, specifically within California waters, with predictions of the water temperature to increase up to 27 degrees Celsius. And this is within some projections from a paper by Wagner et al. In 2011, specifically within the Sacramento River Rio Vista. So to understand the effect of bifenthrin and the additional stressor with increased temperature, Dr. Giroux exposed both fry and juvenile Chinook salmon to several different concentrations of bifenthrin, so 0.15 and 1.5 micrograms per liter for a duration of 26 hours. And they were exposed under 3 different temperature scenarios, 11, 16.4, and 19 degrees. Daily 50% water renewals were conducted, and they were assessed for sex steroid levels, neuroendocrine gene expression, dopamine receptor gene expression, as well as predatory avoidance. So there were different neuroendocrine dysregulations found, and I'm going to focus on the results dealing with the Chinook salmon fry first. Just to orient you with the graphs, we have a relative expression of different genes on the y-axis, and on the x we have bifenthrin concentrations. The 11 degree are denoted in the black box, the 16.4 in the white, and the gradient in the 19. So looking at growth hormone 1 first, we find that if we're just looking at bifenthrin by exposure treatment, we see that there is a temperature effect with significant increases in growth hormone 1 with increased temperature, and we see that across all treatments. And if we compare against increasing bifenthrin concentrations, we do see a trend up with increasing concentrations though not significantly different. With gonadotropin releasing hormone, we find that looking at temperature alone, we see a significant increase in the 11-degree treatment. And if we compare the different concentrations based on bifenthrin, we see that there aren't any significant differences. So there's more significant decreases related to temperature, specifically in this higher treatment group. And with the dopamine receptor, we see, just again looking at temperature differences, we see significant increases related to temperature, though no significant differences compared against bifenthrin treatment, other than this low treatment at 0.15 relative to the control. Interestingly, when juvenile Chinook salmon were assessed with the same exposure concentrations under the same temperature scenarios, actually didn't find any significant change with relation to temperature or bifenthrin exposure treatment in either the growth hormone, gonadotropin releasing hormone, or the dopamine receptor. Looking at the predator avoidance, so this is a olfactory response assay, and a Y-maze assay was used for this. Just to briefly discuss how this works if you aren't familiar. There's an odorant arm and a clean arm, as well as a central arm. So an odorant arm will receive a predatory stimulus. The clean arm will not receive predatory stimulus in clean water. And then the central arm is an area where a fish can just reside without having to choose either or. So we have the percent fish on the y-axis, which shows a specific arm, and either the vehicle control as alcohol or 0.15 or 1.5 micrograms of bifenthrin. We do have two different temperature treatments here, either the 11 or 16.4, but we only saw significant decreases, particularly with regard to the time spent in the odorant arm in the 11-degree treatment, where the higher bifenthrin treatment group had significantly increased amount of time spent in the odorant arm versus the clean arm, with again significant decrease with increasing bifenthrin concentrations of fish that shows the clean arm. In conclusion, environmentally relevant concentrations of bifenthrin under predicted increasing temperature scenarios dysregulated several genes that were within the thyroid and estrogenic signaling pathways in the fry. Bifenthrin, however, may not act through the estrogen biosynthesis pathway in juvenile Chinook salmon at the concentrations used, and possibly through another pathway at this life stage. Increased bifenthrin exposure as well as temperature alter predator avoidance behavior and may be related to impaired olfactory bulbs or altered sensory signaling through a dysregulation of neurotransmitters. So to get a better idea of what was happening within the brain of these exposed fish, we wanted to look at a metabolomic approach. And so we exposed juvenile Chinook salmon to either 0.15 or 1.5 microliters per liter of bifenthrin for 96 hours, and we used non-targeted metabolomic profiles for this assessment. What we found is that overall there was a decreased abundance of several metabolites, specifically inosine, hypoxanthine, and guanosine, with increasing concentrations of bifenthrin. These metabolites were involved or predicted to be involved in pathways that increase the incidence of apoptosis, inflammation, and reactive oxygen species, as well as have predicted effects influenced the axonal growth, outgrowth of neurites, and the branching of axons. The dysregulated levels of metabolites in the brains of bifenthrin-exposed juvenile Chinook salmon were predicted to induce an apoptotic and inflammatory response that may be driven by an increased production of reactive oxygen species, And inhibitions to axon growth and branching in the brains of Chinook are predicted to be related to altered metabolite levels following concentrations of bifenthrin that were detected following stormwater runoff events. However, to look at slightly lower concentrations of bifenthrin, we exposed juvenile Chinook or juvenile steelhead to either 60 or 120 nanograms per liter of bifenthrin for 2 weeks. And again, assessed for non-targeted metabolomic profiles as well as gene expression. We found that there was an altered abundance of metabolites. There were significant decreases in the levels of acetyl-L-carnitine, adenine, and DHA, and significant increases in betaine with increased levels of bifenthrin exposure. Acetyl-L-carnitine, DHA, and adenine were predicted to increase the amount of apoptosis or necrosis within the brains, whereas increased levels of betaine were predicted to significantly increase an inflammatory response. There were dysregulation of genes involved in apoptotic necrotic pathways as well as inflammation. There were significant increases in caspase-3 with increased concentrations of bifenthrin with decreased expression profiles in NRF2 and significant increases in the inflammatory gene myox with increased bifenthrin concentrations. In conclusion, dysregulated levels of metabolites in the brains of bifenthrin-exposed juvenile steelhead were predicted to induce apoptosis, necrosis, and inflammatory response at low bifenthrin concentrations, and genes that were involved in these apoptotic necrotic as well as inflammatory responses validated metabolite profile changes that were following bifenthrin exposure. So to get an idea of some other genes that may be driving some of these metabolomic alterations found in juvenile Chinook and steelhead, we exposed juvenile rainbow trout to either 15 or 30 nanograms per liter of bifenthrin for 14 days, and also looked at tunnel assays for apoptotic cells. We found that at the lower bifenthrin treatment, that gonadotropin releasing hormone signaling pathway was the top predicted altered pathway in the low treatment, and in the higher treatment, there were significant alterations in the extracellular matrix receptor as well as focal adhesion. Looking at the histological alterations using tunnel to again look at apoptotic positive cells. We found that there were significant increases in both the number of tunnel cells in both the cerebellum and opic tectum, with significantly increased concentrations of bifenthrin. Pathways involved in providing cell structure and integrity in the brain were altered following low concentrations of bifenthrin. Gonadotropin releasing hormone signaling pathway, which involved improper neuroendocrine function, was a top altered pathway. An increased number of apoptotic cells in the brain of exposed rainbow trout correlated to altered metabolomic profiles that were seen in juvenile Chinook, salmon, and steelhead trout. So we integrated these two non-targeted metabolomic studies with transcriptomic alterations, and we found that the concentration of triglycerides and overall metabolism of triglycerides were predicted to be affected. And so we conducted a non-targeted lipidomic profile analysis, as well as an additional histopathological characterization of juvenile rainbow trout exposed to 30 or 60 nanograms of bifenthrin. We found that there is a significant increase in the number of congested blood vessels and number of hemorrhages with increased bifenthrin treatment, and also that there was a significant concentration-dependent decrease level of triglycerides with increased bifenthrin concentration in the exposed fish in the brains. And so in conclusion, we can integrate omic profiles to determine common targets of bifenthrin exposure. We can characterize the effect of bifenthrin in the brain of salmonids for understanding the overall influence of additional environmental factors such as temperature. a non-targeted approach for detecting pathways that may be altered, not previously studied. So with that, I'd like to acknowledge the Nimbus Fish Hatchery, Feather River Fish Hatchery, and Just Ranch Lakes for providing the Chinook salmon, steelhead trout, and rainbow trout for these studies, as well as the Proposition 1 grant from California providing funding, and the Metabolomics core facility at the University of California, Riverside for running the metabolomic analysis. Thank you.
Thank you very much, Jason, for that interesting talk. I don't see any questions so far. And in the interest of time, I think we're going to have to move to the next talk. But if anybody has any questions, you can still type them in the question and answer box, and we could get to them later. So the next talk is going to be presented by Noble Hendricks of QEDA Consulting. And he's going to talk to us about a new model looking at occupancy for nearshore fishes. So go ahead.
Hi, welcome to our talk on the approach to estimate occupancy using a novel statistical approach. And we're applying that to some nearshore fishes in the Delta. So first off, we'd like to acknowledge the funding coming from USGS Priority Landscapes Project, and then the U.S. Fish and Wildlife Service personnel who've been involved in collecting the data that we're going to analyze. The amount of time that we've spent in the analysis is much less than the amount of time spent collecting these data, and just want to highlight how valuable their contribution has been. Also want to point out our partners, CDF&W and Department of Water Resources. So the reason for working on this project started from review of the beach seine study, where the limitations of the beach seine in terms of essentially just focusing on beaches and boat ramps was identified, and the desire to want to sample for some of the different estuarine fishes in other habitats. And then on top of that, we want to be able to evaluate or analyze those data using an occupancy framework in which we're explicitly estimating the probability of detection, as opposed to assuming that we are detecting all of the individuals that are in the sampling region. And then furthermore, we're interested in developing statistical designs and sampling designs that will allow us to use space for time replication, which will hopefully minimize the travel time to the sites and allow us to get the information that we need, but at a reduced cost. So a little background on occupancy modeling. The basic question we're trying to answer is a specific site occupied with a species of interest, or is it unoccupied or vacant? And if we can take a sample at that site, and that sample has the species of interest, then we can assume that that site is occupied. If on the other hand, we take a sample and we don't find the species that we're interested in, then we may come to the conclusion that, in fact, that species is not there and the site is vacant. The difficulty with that assumption is that we have as an underlying assumption, and that is that there's perfect detection in that sample. And this is rarely the case. In most cases, we're using gear that's, it does allow fish to escape, or alternatively, we have difficulties in identifying species, even if they're present. And so, for example, we can see here, there's a picture of a flounder. And the point is it's present, but we're not detecting it. And that's coming because either the individual was not captured due to our gear efficiency, or in the case of observational studies, the animal is present, but we're missing it because there's strong ecological reasons or evolutionary reasons for animals not to be detected, most notable of which is that they want to avoid predation. So for our modeling framework, we're going to have two levels. We're going to have a model for the site occupancy. So here we have a probability that the site is occupied. Z is our latent occupancy status. It's 1 if it's occupied, 0 if not. That's equal to mu. And then we have a logistic regression that allows us to relate covariates of interest, which are the x's here, and the coefficients associated with those covariates back to the site level occupancy. And then we have a second level, which is our detection process. And here we have the probability that a sample that we take, or Yij, J being the sample at the site, I, is 1, i.e. we've detected the species of interest. That's a probability, Pij, times the state occupancy. So then we have the probability of detection is a function of covariates that may alter that probability of detection at each of the specific samples. And those covariates are represented here as a U, and the coefficients are gamma. One thing to note here is that we only detect individuals at a site if that site is occupied, or another way of putting that is there's no false positives. So for example, the types of data we might collect for occupancy analysis, if the site is occupied, then we might have samples, for example, we detected, we didn't detect, and then we detected. So this would be Yij 1, 0, 1. We could have a situation where the site is occupied, but we don't detect it in any of the samples. And that might occur if our probability of detection is low under the environmental conditions at that site and time. And then the last is if the site is unoccupied, then the only result that we can obtain is non-detections or zeros in our Yijs, because again, we're assuming that there's no false positives. And there are other occupancy models that can deal with false positives. They just require a little bit more in the way of the dynamics. So one of the things that's really important for the occupancy modeling framework is the need for replication. And the ideal replicates would be IID, that is independent and identically distributed. So they would give us information on that probability of detection given occupancy. Generally, in the framework as it was originally developed, the replication occurred temporally, and that was by returning to sites over a short period of time between those visits, site visits, and collecting the presence or absence of certain species. The difficulty, particularly in working in the Delta, is that the temporal replicates can be expensive because the largest component of the cost of taking the sample is getting to the site. So spatial replication is really attractive in that multiple locations can be sampled at a specific time or visit, and then those can form the replicates. However, the rub is that the spatial replicates may not be independent, and in particular, they may be spatially correlated. And the closer the segments or the samples are together in which you're taking those samples, the more likely we're going to have spatial autocorrelation. So in our case, we're motivated by wanting to develop a series of samples in which we're using shoreline spatial replicates and electrofishing as our approach for collecting fish in the Delta. So for example, we have here, hypothetically, 7 contiguous segments where we will be sampling, and the fish that are identified and collected in each one of these segments will be each one of our replicates. And our task now is to be able to develop a statistical approach that can incorporate in this segment level spatial autocorrelation. So here is the sampling design as it's currently being implemented. We have 6 segments, replicate segments located adjacent to each other. So we're expecting spatial autocorrelation. And the role of the spatial replicates is that now we need to evaluate the segment level occupancy status. So with a space for time replication design, this becomes a new layer in our model. So we now have segment level spatial, the segment level occupancy. So is the segment occupied? That's going to be a latent state, just like it was for the site level occupancy. And then the last question is the species of interest detected, given that that segment is occupied? So because we don't have replicates at the segment level, we need to do some structuring of those segment level occupancy status. And the approaches that were first developed for structuring the segment level occupancy used a Markovian approach, which basically means that the segment before, the sample in the segment before then gives you information on the current segment you're interested in. So in our case, we're interested in segment 3, the dynamics that are occurring. So we'll call this our focal segment. And the approach is that segment 2 tells us a little something about segment 3. So we expect this type of spatial correlation. And this model was originally developed for tracking tigers on trails and using paw prints and scat as the indicator of presence for the tigers. So for large mammals, this may make sense where there's a directional movement along a segment. And the approach was then to use 2 probabilities. So if the segment before is occupied, then there's a probability that the current segment is occupied. That's got a probability theta. And then the other alternative is if the segment before is not occupied, then there's a different probability that the current segment is occupied. That's theta star. And then using theta, 1 minus theta, theta star, 1 minus theta star, we can cover all of the possible transitions that could occur. The issue with this approach is that only the previous segment is being incorporated into the modeling paradigm. And we would actually like to be able to have both neighbors or multiple neighbors be incorporating into the probability of segment occupancy. So for this, we're going to use an autologistic regression approach. And the autologistic essentially allows us to be able to define this focal segment, 3 again, and incorporate in our neighbor to the left, neighbor 2, and our neighbor to the right, segment 4. And this is a type of regression, type of logistic regression, where the responses are not independent. And in particular, they're incorporating in these neighborhood effects. So biologically, we have the potential for an aggregation process that's occurring within our species of interest. And that shows up essentially as local hotspots. And we want to be able to capture those dynamics within our segment level occupancy. So we structure the segment level occupancy using this autologistic regression. So we have a term in this model called, that's lambda, and that's the strength of this spatial autocorrelation. And if lambda is equal to 0, then we have an independent case. And if lambda is positive, we have an aggregation process. If lambda is negative, then we have more of like a repulsion process. So we apply this work in this model to 3 species of interest: Chinook salmon, largemouth bass, and hitch. So we have a seasonal species, a introduced species with wide distribution in the delta, and we have an endemic species that's somewhat rare currently. And then we're going to look at the probability of occupancy by month and region. We've also included some probability of detection covariates, although we won't look at those within this talk. And then we're fitting this model using JAGS, just another Gibbs sampler, and a Bayesian framework. And we will also be estimating this lambda parameter, which is the strength of the autocorrelation. So here's the posterior mean predictions for occupancy. So for Chinook, we see that there's dominant or probabilities of occupancy are highest in the North Delta and in April. For Hitch, we can see that the probabilities of occupancy are increasing over the spring and are highest in Cache flu area. And for largemouth bass, we've had to rescale. So notice the scales here are slightly different. They're pretty much ubiquitous with higher probabilities, close to 1, in both the South Delta and Central Delta, and then not so much in the way of variation in the temporal structure. So they tend to be in those habitats year-round. So now if we look at the spatial correlation coefficient, theta, lambda, sorry, what we see is that for all species, the values are positive. So a value of 0, again, would indicate no spatial autocorrelation. And we can see that it structures such that Hitch and bass have higher aggregation or spatial autocorrelation relative to Chinook, but they're all positive, indicating that the model is picking up on the spatial correlation in those segment level occupancy dynamics. Thank you very much for listening and watching in the talk. If you have any questions, we have a short question and answer session, but if anything comes up after that, please contact me at the email address here. And thanks again for watching the talk.
Thank you, Noble, for that interesting look. That's a clever approach to a real sticky problem. See if we've got any questions here. We have a couple minutes for questions. If anybody has any, you can use the raise hand function. Well, Noble, I have a question, if I may. Given this approach you've developed, how does it compare to what was done previously with these data?
Yeah, so can you hear me okay, by the way? Yeah. Yeah, so we get similar estimates, I would say, of the probability of occupancy. Those patterns shifted slightly, but not dramatically. But I think the patterns or the sort of the relationships and the probability of detection is where we've seen a little bit more of a change. And it's because of the that local occupancy at the segment level that's being driven or related to that lambda parameter and the role of the two neighbors. That's really affecting that detection process, or it's involved more in that detection process than it is in the occupancy at the site level. So that's where we're seeing a little bit more variation in the two methods.
Okay. And just to follow up, I noticed there was more likely to have occupancy with the species with a largemouth bass compared to like the Chinook. And do you think, is that a function of just the behavior of the fish? Like the largemouth might be more stationary, or whereas the Chinook might be migrating through?
Yeah, I think that's right. I think that the occupancy models are picking up on that temporal component of the Chinook. Actually had sort of that same question to ask Fred, which was if they've done any tagging of some of the fish that they were capturing to see how long they're residing in those different reaches, because that's kind of the picture we're seeing. Although I think, yeah, and it would also be interesting to see kind of how the size ranges overlap with the fish that we're picking up in this electrofishing study relative to the sort of more predator focused that he's got.
Okay, well, thanks a lot, Noble. That's really cool work. We'll move on to the last.
We actually have a question in the audience. Walter Griffiths, I believe, had his hand up.
Oh, I'm sorry, I didn't see that.
Walter, you want to raise your hand again?
Christine, are you able to unmute him and allow him to ask the question?
Yeah. Go ahead, Walter. You should be able to unmute.
Hi, Noble. Thanks. Just playing with this idea in my head. The lambda parameter, the strength of the aggregation. Could you use that potentially to, I don't know, make recommendations for, like, ideally how far apart sampling needs to be truly independent, or is that something that would be of interest in a model, applying a model like this?
Yeah, that's a good question. And I think what we would want to do is maybe look at the neighborhood structure. So right now, the way the neighborhoods are structured, it's just, you know, the one cell or segment on one side and one segment on the other. But we could expand that and make it 2 on either side, and basically look to see how those lambda parameters vary by changing the neighborhood structure. And we might be able to obtain a little bit of information there on how, you know, basically how that, because it should attenuate, I think, as the neighborhood structure gets larger. And then that would give you some idea of what that, you know, at what point really you're not, you know, there's no incorporation of new information from the neighborhood.
Neighborhood in this case means the length of the electrofishing.
The neighborhood would be the number of segments either side of the focal segment that you were incorporating into that autologistic function.
Okay, great. Thank you, Noble. We're going to have to
Follow up.
That doesn't make sense. Follow up later. Move on to our last talk, which is going to be a team effort given by Emily Funk from the University of California, Davis, and Melinda Bearwald from the Department of Water Resources. And they're going to tell us about how to identify the different runs of Chinook salmon using a CRISPR technique. So go right ahead.
Hello, I'm Emily Funk from the Genomic Variation Lab at UC Davis.
And I'm Melinda Bearwald from the Department of Water Resources. And today we will present on how rapid CRISPR-based genetic identification of run type enables new opportunities for the management of listed Chinook salmon. We would like to acknowledge our co-authors and thank them for their contributions. California's Central Valley is at the southern edge of the range for Chinook salmon, yet contains the most diverse assemblage of the life history types found in the world for the species. There are 4 runs, each named for the season of its upstream spawning migration. All of the runs have declined in abundance from historic levels, with Spring Run experiencing the most dramatic decline, and it's listed as threatened under the federal and state Endangered Species Acts. Winter Run was never an abundant run type, but it is found nowhere else in the world outside of the Central Valley. The population size has declined dramatically, and it was given ESA endangered status. The other 2 runs, Late Fall and Fall, are considered species of concern due to hatchery influence and decreased population sizes. As you can see from the graph, in recent times, Fall Run is by far the dominant run in the Central Valley. So if you just count Chinook salmon in the Central Valley, you'll get a sense for how the Fall Run population is doing, but not gain insight into what is occurring for the other more vulnerable runs. Therefore, it becomes critically important that we can assign Chinook to their runs. Some of the reasons why include so that we're accurately monitoring population abundance and distribution throughout the system, getting accurate counts of listed species take at salvage that can impact water operations, and can evaluate the effectiveness of restoration activities or other management actions intended to benefit a particular run or runs. Since there are no morphological differences between the runs, other approaches needed to be devised, and the longest running, it's been around since the 1970s, and to this date, most common method for run type identification of migrating juveniles is termed the length at date criteria. The great thing about length at date is how easy it is to do. You just need to know the date you captured the fish and its fork length. The bad news is that multiple studies have now shown that underlying assumptions to length at date aren't true, and that there is considerable overlap in distributions of run-specific fork lengths of genetically identified individuals. Looking at some actual data, in the little box, you can see the color assignments for a particular length at date run type. And then the main bar graph plot is showing you the genetic assignments and how the length at date assignments partitioned out across them. Taking you through a quick example, for individuals that assigned to spring with the length at date criteria, only 31% of them also genetically assigned to spring run. So the majority of these individuals genetically assigned to one of the other runs. The inaccuracies happen in other ways as well. As I said, this is our most common way of determining run types. If we're observing these inaccuracies when using this tool, then how can we have confidence in any downstream data that relies on these run type assignments? I'm a huge fan of easy, but we need easy and accurate to make the best management decisions for the species possible. A promising alternative to the original length at date approach has been developed by Noble Hendricks for winter run, and he's now beginning development for spring run probabilistic length at date, or PLAD, models. These are site-specific models that rely heavily on genetic data and associated metadata to build the initial PLAD curves. Additional genetic data may be helpful in some situations to better parameterize models. A key difference between the PLAD output and the original length at date criteria output is that PLAD models can assign more than one run to a given juvenile Chinook salmon. This enables one to create thresholds in terms of the probability of assignment required for you to trust that result. We'll have a very good feel for how accurate each site-specific model is because we can test it by comparing it to the genetic results of samples that weren't used to create the model curves. Testing tools for their continued accuracy is something that needs to happen iteratively, and that is the plan.
So to inform the PLAD models, we need genetics, which can accurately identify run type and in some cases tributary of origin. Ideally, the genetic method is accurate, sensitive, fast, low cost, high throughput, easy to use, and won't require expensive specialized equipment. No method is perfect to answer all of the genetic questions while at the same time giving you all the characteristics I just listed. We seriously considered four genetic methods for one project, the juvenile production estimate, and two methods are being further evaluated and compared at the same time, GT-seq and CRISPR-based assays called Sherlock. There are advantages and disadvantages to all genetic methods. qPCR and Fluidigm are mainstays of genetic identification, whereas Sherlock and GT-seq are more recently developed. The benefit of Sherlock is that it gives a fast field deployable run type ID. However, Sherlock only targets one SNP or indel per assay, and for Chinook we only have 4 assays, whereas GT-seq frequently uses hundreds of SNPs. So GT-seq may be the better method for assigning individuals to tributary. Sherlock has the potential for running many assays, but we currently don't have that system up and running. It's possible for the future if that's something we want to pursue. And today we'll be talking about the development of Sherlock assays for Chinook salmon run type ID.
Before discussing Sherlock in more detail, we want to mention that PLAD and a genetic approach such as Sherlock are complementary and can be used synergistically together for run ID assignment that optimizes accuracy and efficiency. We're piloting their combined use for the Juvenile Production Estimate Spring Run ID program. One of the conditions of the incidental take permit is to develop and implement a juvenile production estimate for spring run Chinook salmon. We therefore require accurate run IDs for all collected individuals. The idea is that PLAD can be used as an initial screening tool for all samples to improve efficiency. Using this juvenile production estimate as an example, if the R&D phase showed that the spatial model was highly accurate and an individual assigned with high probability to fall or late fall, then nothing more needs to happen to that sample. It's identified. As you'll recall, fall run is by far the dominant run in the system. So a lot of samples could go down this path if we have confidence in the spatial models. However, if a sample has a low probability of assignment to a single run type, or there's a low confidence, or it is identified as a spring or winter run sample, then it moves forward to be genetically verified. Results from the genetics are fed back into the PLAD for near real-time model improvement throughout the season. We're just beginning this combined approach this year, so we don't have any data or results at this time, but we think this has really great potential, particularly the closer you are to the spawning sites. For now, though, we'd like to dive into some of the more important details about what SHERLOC is and what it can do.
So what is SHERLOC? SHERLOC is a CRISPR-based technology, CRISPR standing for Clustered Regularly Interspaced Short Palindromic Repeats. And most people think of the Cas9 gene editing technology when they hear CRISPR. However, there are many other Cas enzymes that have different functions. Cas13 is an enzyme with RNase activity that can be used for RNA knockdown, RNA editing, and nucleotide detection. The nucleotide detection function of Cas13 can be used to signal the presence of a particular nucleotide sequence, such as a diagnostic SNP, which can be used for run identification. So SHERLOC stands for Specific High Sensitivity Enzymatic Reporter Unlocking. And to perform a SHERLOC assay, the first thing you need is a sample for your putative specimen of interest. This could be diverse material, including blood, thin tissue, mucus, or even eDNA, environmental DNA. Next, RPA, recombinase polymerase amplification, is used as an isothermal alternative to PCR, so the entire SHERLOC reaction can occur at 37 degrees. If using a DNA input, the amplification product is then transcribed into RNA. Then, if Cas13 finds its target sequence, it goes into overdrive, cutting RNA, but not just your target RNA, it also cuts other RNA that's in your reaction. So that collateral cleavage can be used to our advantage by including an RNA reporter in the system that once cleaved gives off a fluorescent signal. And that only happens after the CRISPR detection of your target sequence. And just to note, this entire process happens in a single tube. We did a lot of development and testing to optimize this approach to identify the different runs of Chinook salmon, and I'm now going to present some of our results. First off, there isn't a single SNP or indel that discriminates between all 4 run types, so we took a tiered approach. So first, we distinguish between early migrating phenotypes, which are spring and winter runs, and late migrating phenotypes, which are fall and late fall runs. These 2 SHERLOC assays are specific, meaning that the early migrating assay detects only spring and winter individuals, and the late migrating assay detects only fall and late fall individuals, as shown in these heat maps. The light yellow is no detection, and the dark blue is full detection. On the left, we're showing specificity by run type, and on the right, we're showing specificity by tributary. If you notice, the greenish blocks in these heat maps, those are tributaries that contain a few heterozygous samples, which were detected by both assays. And it is known that hybridization can occur between the different runs, and these tributaries with the heterozygous samples are known to contain multiple runs of Chinook salmon. We also tested the sensitivity of these assays and found that we can detect down to 6 DNA copies or fewer per reaction, which is on par with the sensitivity of qPCR, which is considered the gold standard. So the second tier is to distinguish between spring and winter runs. So if a sample identifies as an early migrating phenotype with the first set of Sherlock assays, we then perform a second set of assays to identify winter or spring run. At this time, we don't have a second set of assays to discriminate fall from late fall because they are genetically very similar, and it was also lower priority at this time because they are not ESA listed. Our spring and winter assays are run type specific as shown in these heat maps. The winter assay identifies winter individuals and the spring assay identifies spring individuals. We also ran a time course experiment with our early and late migrating Sherlock assays to test the speed of the reaction. We're trying to make the assay as fast as possible to acquire near real-time results. As you can see, the signal starts exponential amplification at 20 minutes, and we can make an accurate ID call by 25 minutes, shown by the red line in the graph. And each of these lines on the graph are a run type. We've also optimized this assay for use with mucus DNA so that, which does not require a DNA extraction, further speeding up the time to results compared to other standard genetic methods, and making it even more field deployable. We've produced a video and SOP that are available showing the swabbing technique so that field biologists can pull it up at any time and review it.
To perform Sherlock in the field, you'll need minimal supplies as shown here. Sample collection tubes and swabs for obtaining mucus, strip tubes containing the reaction mix, and a cooler for keeping the mixture frozen until use, as well as a portable fluorescent reader to run the reactions, and gloves and a data recording sheet. There is also the possibility of using lateral flow strips instead of a fluorescent reader. These lateral flow strips are like a pregnancy test. You dispense the reaction onto the strip and get a colorimetric line. As shown in this figure, the strip on the left has 2 lines indicating a positive test, and the one on the right with 1 line is showing only the control band. While we have a lot of other things in the works, we want to finish up with a new collaboration we recently began with the Monterey Bay Aquarium Research Institute, or MBARI, to add Sherlock into their existing environmental sample processors, or ESPs. They take these ESPs all over the world and collect water for weeks to months at a time in any type of water body. The ESPs can be stationary, such as the one shown on the left, or they can be housed in mini submarines, which can be remotely controlled. Inside the housing are filters, which can collect water for eDNA studies at chosen intervals. A super cool feature is that an ESP can also perform molecular reactions, such as a qPCR or even sequencing, and can send the results remotely to shore. We're working with MBARI to bring Sherlock technology on board so that the molecular reaction will be quicker and more stable for long-term deployments. This technology could be super useful for passive monitoring in the Bay Delta for continuous and real-time detection of listed species, new invasives, and nuisance organisms such as harmful algal blooms. Early days for this collaboration, but we're excited to see where it goes. And with that, we'd like to thank everyone that's contributed to this project as well as our funding agency. Thanks so much for listening. Any questions?
Wow, thank you, Emily and Melinda. That is some pretty cool technology. Check the Q&A. We got any questions, or does anybody have any other questions? They can use the raise hand feature. Not seeing anybody with their hands raised yet. We've got like a minute and a half. Okay, we have a question from Tyler. Okay, Tyler, go ahead.
sorry, I have to click the unmute button. I had a question about those test strips. Are those, what is the typical cost on those? And can you, I'm guessing you could get them with various different primers associated with them or for different species?
Yeah, so the cost for them, they cost usually a few dollars, like maybe $3 to $5 range for the test strips. So they're more expensive than the fluorescent assay itself would be. But in terms of what type of species they can do, any type of species whatsoever. And so it's the test strips have streptavidin coating on them. And so you design the assay and then it is then the band gets produced when your particular assay binds with the streptavidin.
Great, thank you.
Mm-hmm.
Okay. Well, it's 2:40 now, so it's time for the 10-minute break. I'd like to thank everybody for attending this session, and thank you to all the speakers for the really great talks we've had. And with that, I'll say thank you and enjoy the break and stay tuned for the next session.